Now, as we talk about the membrane, let's look at some of the important functions of the membrane. It is going to allow exchange of material. So while we have a separation between the ICF and the ECF, we also have exchange happening between those two mediums. It's also going to be selective again in that some things can cross and some things can't. So it's going to decide where certain properties of certain molecules are more favorable to go through that membrane and other things are not. So we'll talk about things like large, bulky particles. They cannot get through the membrane. Charged particles like ions, they're going to need transporters or ion channels to get through that membrane. They are not going to be able to freely pass through because of the membrane structure. Some of the things that we do want to be able to get through that membrane are oxygen and certain nutrients, as well as our waste products and byproducts, right? Things like carbon dioxide, ammonia, which are some of the byproducts from cellular respiration, right, which we talked about. Those things need to be able to get out of the cell easily, and so they are exchanged for more important things like oxygen, which we know is going to cellular respiration, and then nutrients like glucose, which we know is also going to be metabolized. So the exchange that happens across the membrane is really important to an overall function of the cell. Now, that selective permeability means that some things can go through, other things cannot. The nonpolar items, nonpolar molecules will easily be able to transport through that membrane. So oxygen is nonpolar, carbon dioxide is nonpolar, fatty acids are nonpolar, and they need to be able to freely move through the membrane. Ions are in polar molecules, so sodium chloride, large bulky proteins, glucose. Those things are going to need transporters in order to be able to get through the membrane. All right, let's move on to the second objective here to explain the role of chemical, electrical, and electrochemical driving force in the passive transport of substances across the membrane. And then we're going to distinguish between passive transport and active transport. Okay, so as we think about passive versus active transport, we want to sort of visualize this as the direction of movement. So passive transport is going to be a downhill movement. Think about a ball on the top of a hill, and as it moves down the hill, that is synonymous with a passive type of movement. In other words, that's going to be spontaneous. That's going to happen sort of organically. That's going to liberate energy as opposed to requiring energy. So passive transport is a downhill movement. It happens spontaneously in that it doesn't need any energy inputted for this to occur. This will occur naturally or spontaneously, and that will be a downhill direction, which is giving up energy. Active transport, on the other hand, is if we have a ball at the bottom of the hill. Think about this green ball here, and we want to move that to the top of the hill. So this is not going to happen spontaneously, right? This is not going to just happen naturally. This is going to require energy input, and this is going to be an uphill type of movement. So to get that green ball to the top of the hill is going to be an active transport. To get the orange ball to the bottom of the hill will happen more spontaneously, and that will be passive transport. Okay, now we've described the structure of the membrane. We've talked about the different types of transport. And here we want to start putting that all together by talking about our chemical and electrical forces. These are the forces that are acting on particles to get them to move across the membrane. Now, when we combine both of those forces, this is the electrochemical force. And this is basically a sum total of the two individual forces that are also acting on a particle. Now, this can encourage the particle to move downhill, which is passive, or to move uphill, which is active, right? And so as you begin to look at these two forces, we will talk about them individually. So we'll talk about the chemical force, and then we'll talk about the electrical force. And then we'll better be able to understand them as separate forces. And so we can combine them and think about the overall sum electrochemical force. All right, so let's begin by looking at the chemical driving force. And so the chemical driving force is based on the concentration gradient, which is the difference in particle concentration across the membrane. So if we have more of one particle on the outside of the membrane, and we have less of one particle on the inside of the membrane, automatically we have a gradient, okay? And that gradient wants to equalize those particles across the membrane. So that gradient is going to want to push the particle from higher concentration to lower concentration in order to equalize the concentration across the membrane, okay? So this is a force, right? This is what we mean by a driving force. It's an energy or an influence that wants to move the particle. So the chemical driving force is created because there's an unequal concentration across a specific membrane. All right, so let's illustrate that here. Okay, so the chemical driving force is basically the movement of particles down their concentration gradient. All right. The force that's moving the particles is going to be from high to low concentration, right? So it's a force moving particles from high concentration to low concentration. Okay. So let's see what that actually looks like. So let's say we have a cell here. We know we have this cell membrane, which these particles have to be able to get through. So it's separating two different environments. On the outside of the cell, we have a lot of particle A. Right, so let's draw this here, particle A. And on the outside of the cell, we've got lots of particle A. Okay, let's also label that this is a semi-permeable membrane, right? So this is a semi-permeable membrane. So in other words, while these particles are being separated, they could get through if the conditions allow, right? If there was enough of a driving force. So on the inside of the cell, we have very few numbers of particle A. Let's look at particle B. So here is particle B, which is in blue, and we have the opposite concentrations for particle B. So we have a lot of particle B on the inside of the cell, in other words, a high concentration of particle B on the inside of our cell, and then a lower concentration of particle B on the outside of the cell. Okay, so we can already begin to describe what the driving forces would look like. So who can tell me the direction of the driving force for particle B? What direction is the chemical force for particle B? Where is particle B going to want to move? Perfect, yes. So to the extracellular environment or out of the cell. Excellent, thank you. So particle B, high concentration on the inside, low concentration on the outside. Particle B is going to want to move to the outside of the cell. So we're going to draw this large arrow here to represent the chemical driving force that is acting on particle B, okay? As a result, right, of what we just described, particle A is going to want to move in the opposite direction. So because there's a higher concentration of particle A on the outside, a lower concentration on the inside, particle A wants to move in the opposite direction. So here is this large arrow showing the chemical driving force that's moving particle A. So this is what we mean by the chemical driving force. And if we think about drawing what that looks like, right, we want to remind ourselves that particle A is moving down its concentration gradient. So here's the outside of the cell. Let's draw this. Here's the outside of the cell, right? Here's the inside of the cell. As particle A moves from the outside to the inside, it's moving down its concentration gradient, right? So this is going to indicate the direction of movement down its concentration gradient as it moves from the outside to the inside. And the opposite is true for particle B. As particle B moves from inside of the cell to outside of the cell, it's also moving down its concentration gradient. Okay, so while we think about it moving from the one direction to the other, also think about that type of movement as it relates to the passive transport, right? It's going down its concentration gradient. Naturally, this is a spontaneous type of movement as it moves from a place of high concentration to a place of low concentration. Okay. All right, let me just give another few seconds to finish up, and then we will continue. Okay. All right. Now, we can also think about the electrical driving force as a separate force. As far as the terminology here, we want to think about some terms that will help us understand the electrical driving force. So an ion is any type of charged particle. Specifically, a cation is a positively charged particle, and then an anion is a negatively charged particle. So we can think about sodium as being an ion, right? We sometimes refer to these as electrolytes because, again, they're charged. So if it's positively charged, this is a cation, like sodium is. If it's negatively charged, like chloride is, it will be considered an anion. We can also describe the membrane potential. And the membrane potential is represented by this large B and then a subscript M. And what this is it's a force. And the description of this force is that it is caused by the unequal distribution of our charged particles. So because we have more sodium on the outside and more potassium on the inside, also because of our cell membrane, right? Remember, our cell membrane separates these two fluid compartments such that we can have a different distribution of ions happening on the extracellular fluid, and we can have a different distribution of ions happening on the intracellular fluid. So because of that separation, that unequal distribution of the charged particles, we now have a force that wants to move those particles, again, with an attempt to equalize their distribution across the membrane. So the charge separation is a source of energy. It's another force that wants to move these particles to help equally distribute these charged particles. So again, if you think about sodium, there's more sodium on the outside of the cell, which creates more of a positive charge on the outside. There's less sodium on the inside of the cell, which creates a negative charge on the inside. And there we have a driving force. It wants to move sodium from a positively charged area. It's attracted to that negatively charged environment, and so it wants to get on the inside of the cell. Now, if we think about the magnitude of the membrane potential, That is describing the strength of the force. So how strong do these particles want to move? How much do they really want to get across the membrane? And this is usually measured in millivolts. And with this, we want to think about the cell as having a polarity. So the inside of the cell is more negative than the outside of the cell. Again, because of that unequal distribution of the charged particles. So a positively charged particle like sodium is going to want to get to the negative environment that exists on the inside of the cell. A negatively charged ion like chloride is going to want to get to the positive environment that's existing on the outside of the cell. So that polarity is what we describe as being the strength of the membrane potential. And when we refer to that value, we're usually referencing what's happening on the inside of the cell. So if I tell you that the membrane potential is negative 70 millivolts, what that means is that referencing the intracellular fluid, the inside of the cell is a negative 70 with respect to the outside of the cell. Okay, if I tell you that the membrane potential is 100, right? It's positive 100. That means that the inside of the cell is positive 100 with respect to the outside of the cell. So when we talk about the membrane potential value, it's always from a reference point of the intracellular fluid and not necessarily the extracellular. All right. So let's look at what that would actually look like. So here's our plasma membrane separating the intracellular fluid here on the left from the extracellular fluid here on the right. Now, again, notice the ions are not equally distributed. We have an abundance of cations on the extracellular surface, and we have an abundance of anions on the intracellular surface. So therefore, we create a positive environment in the extracellular fluid and a negative environment inside the cell in the intracellular fluid. So now we can begin to think about what that creates in terms of a force for charged particles to want to move to be in either of these environments. So a positively charged ion like sodium is attracted to the negative environment that's in the inside of the cell. A negatively charged ion like chloride is alternatively attracted to the extracellular fluid, which is overwhelmingly positive. So the separation of charge, the unequal distribution of the charge is what creates the electrical driving force. And that is a reflection of the attraction of negative to positive, right? The attraction of ions wanting to move to be in their environment that is of opposite charge. Okay, let's continue here describing the electrical driving force. So the principles that govern the electrical driving force, again, is that opposites attract, right? So opposite charges want to attract to each other and like charges repel. So I keep going back to that sodium example. Sodium is positively charged. It doesn't want to be in the extracellular fluid. It wants to really get into the inside of the cell because opposite is going to attract and like charges will repel. The other important principle is that the direction of the force depends on the polarity of the cell, which is created by that cell membrane, the fact that the ICF has a different polarity, a different charge than the ECF, right? As well as the charge of the particle in question. So the direction of the electrical driving force will be determined by the negative and positive polarity of the cellular environments, ICF being negative, ECF being positive. And the other thing that determines the direction of this force is the charge of the individual particle that we're speaking about. So different particles have different charges. If we think about potassium, potassium is going to want to get to the negative environment. Sodium is going to want to get to the negative environment. Alternatively, negatively charged particles like chloride will want to get to the positive environment. So those two variables will be important in determining the electrical driving force. Okay, so we'll do a last sort of review here of the electrical driving force, again, to illustrate kind of a little bit what that looks like and what that means here. So this is going to depend on one, the charge on the particle in question. And two, the polarity of the cell. Oops. The polarity of the cell. And when we say polarity here, we mean the difference in the charge, right? The state of the cell. Whether that be positive or negative, okay? So let's look at two cells, and we're actually going to look at two different examples because I want to look at what a cell looks like at rest. So here's a resting cell. And then we want to think about what a cell looks like if it's excited, okay? So here is an excited cell. And so this is going to be really important to form the basis of what we describe when we begin to talk about the exciting of a cell, the membrane potential, the action potential, etc. So on this cell, we've got a lot of negative charge on the inside of the cell, overwhelmingly negative charge inside the cell, and then we've got less of a negative environment outside, okay? And then we can think about an ion like, actually, let's not do that yet. Let's look at the positive charge. So alternatively, we have an overwhelmingly positive charge on the outside of the cell, and then a lesser charge on the inside. So if we think about an ion like sodium, sodium is going to want to get to the inside of the cell, right? So sodium is going to want to move from an overwhelmingly positive area to the negative environment inside the cell. So here's the direction of sodium's electrical driving force, okay? Alternatively, if we think about chloride, chloride is negatively charged. So chloride doesn't want to be on the inside of the cell. It wants to move to the overwhelmingly positive area on the outside of the cell. So here is the direction of the electrical driving force that would be acting on chloride, okay? Now, the last thing I want to say here is that this is the cell at rest. A resting cell has a membrane potential of negative 70 millivolts, right? Negative 70 millivolts. And as we described earlier, sorry, millivolts should be here. Let's do that again. And as we described earlier, negative 70 here means that there's more of a negative environment, right? And the strength of that being 70 on the inside of the cell than there is on the outside of the cell. So that's what that negative 70 millivolts means. It's always referencing the intracellular fluid as opposed to the extracellular fluid. Now, when we excite a cell, we are going to increase the membrane potential value to positive 30, right? And what that means is that the orientation here is going to be reversed. So here we have lots of sodium, or lots of positive charge rather, on the inside of our cell, right? Notice the inside now is overwhelmingly positive. The outside, lesser so. And then the outside is, oh, oops. The outside is overwhelmingly negative, right? Abundance of negative charge on the outside here. And then the intracellular fluid, lesser so, okay? So if we think about the direction of our particles in an excited cell, it's going to be the opposite. An ion like chloride is going to want to get from the positive environment, or excuse me, from the negative environment outside, the positive environment on the inside, right? So chloride really wants to get to the overwhelmingly positive area. And the opposite will be true for sodium. Sodium really wants to get to the negative environment on the outside of the cell. So the direction of sodium's electrical driving force for an excited cell would be on the outside.